Today we want to talk about T-cell immunity and its relationship with COVID-19. Since there's a lot to talk about in immunity itself, we'll be giving a simplified version of how immunity works. Although we can simplify immunity, I think one thing that we can all agree on is that the pandemic itself is not simple. With the daily death rate rising because of the advent of flu season, our situation is complicated. But it is not unsolvable. If we're going to defeat the coronavirus, then we're going to need to know how it works. Let's dive in. Now, the two main proteins that we want to focus on here are the nucleocapsid protein, or N protein, and the spike glycoprotein, or S protein. Both of these serve integral roles to the virus, and will come later when we start analyzing our data. Now, these proteins are also of interest to us because they are very similar to the proteins found on other coronaviruses. For example, SARS-CoV-1 that caused the 2003 SARS outbreak, or the coronavirus that caused MERS, or Middle Eastern Respiratory Syndrome. All of the viruses shown here demonstrate a high similarity to SARS-CoV-2, a fact which researchers did and are still using to learn more about SARS-CoV-2 itself. Now, there are two ways that the immune system protects our body from pathogens like SARS-CoV-2. We'll mostly talk about adaptive immunity, specifically the pathway that activates T cells and B cells. Our narrative starts whenever a pathogen enters the host body. For example, let's say a bacteria. We start with the dendritic cell, which actively tries to find dead bacteria and collects their antigens, or molecules that are produced on the surface of a pathogen, which the immune system uses to identify that pathogen later and also to activate different kinds of immune cells. Once the dendritic cell has its antigen, it travels to the lymph nodes, which is filled with naive or unactivated T cells and B cells. Once the dendritic cell presents its antigen to a T cell or a B cell, it will activate. Now, it's ready to fight against the infection. Let's start with T cells. Whenever a T cell is activated, it'll differentiate into three main types, which all serve different purposes. These three types are then also separated into either memory or effector cells, with the memory cells lasting longer and staying after the pathogen has already gone, and the effector cells only lasting while the infection is still prevalent. One of the effector cells is the killer or cytotoxic T cell, which commands all infected cells in its vicinity to commit cell death or apoptosis. Then you have its friendlier brother, the helper T cell, which activates other naive T cells and, most importantly, the B cells. Now, when the B cells are activated, they will create antibodies tailored to the specific pathogen that is invading the host. These antibodies kind of act like homing missiles that will track the antigen on the surface of the pathogen they're tailored for. The antibody will stick to the surface of the pathogen, both hindering the pathogen's movement and also making it easier for other immune cells to kill off the infection. Last but not least, we have the memory T cell. Now, this is one of the most important mechanisms that the immune system uses to maintain long-term immunity, and also to mount up a good immune response whenever a host is reinfected with a dangerous pathogen. Its job is to essentially remember the immune response that occurred whenever a pathogen first entered the body, and then replicate that. This allows the immune system to build up a response in a much faster time than would be without the memory T cell. Now, the reason why we're focusing on the T cell so heavily is because we found several studies that imply the importance of the T cell. A paraphrase from one of our studies says, "A failure to develop immunity could occur due to an insufficient T cell response, given that the antibody, or the B cell response, is dependent on helper T cell, or CD4 T cell, activity." Now, given this information, we can talk about the different degrees of T cell immunity. For example, if we have only a moderate immune response to the virus, then we might experience mild reinfection and also the chance of asymptomatic spread, or if we have a robust immune response, we may actually end the pandemic.
Maybe 80%.
Now, one of the ways that researchers have been trying to figure out how to prompt a good immune response is through looking at prior immunity. Take this figure, for example, that takes helper T cell reactivity to the spike protein. This is similar to the other figure, which also looks at T cell reactivity to the N protein instead. Now, both of these indicate that there's T cell reactivity during the pandemic and also pre-pandemic, which implies that there might be some sense or some presence of prior immunity.
Given the evidence of prior immunity, this may go on to link T cell memory to the vast number of asymptomatic infections we see today. According to the CDC, 20 to 75% of the general population may be asymptomatic to COVID, which may correlate with an appreciable T cell reactivity to COVID prior to infection. In one study, patients who recovered SARS 2003 continue to show T cell reactivity to the nucleocapsid protein on SARS-CoV-2, suggesting that T cell immunity in coronaviruses or immunological memory in general may last at least 17 years. So in this next graph, the red bars indicate T cell reactivity and the yellow bars indicate seropositivity, which is the presence of antibodies in the blood. This study found 25% T cell reactivity in blood samples collected in mid-2019, suggesting prior T cell immunity to the virus. Almost 50% of supposedly unexposed individuals showed a T cell reactivity to the virus after the pandemic, suggesting a higher rate of asymptomatic infection than previously known. Throughout this figure, you can see that the seropositivity is significantly lower than the T cell response, which suggests that antibodies to COVID may wane over a short period of time. But in another study, they found that individuals with prior immunity to the OC43 seasonal coronavirus who had cross-reactive antibodies to SARS-CoV-2 had no protective immunity compared to people who did not have any prior immunity to OC43. In these graphs, they are comparing COVID antibody titers between people who had OC43 and got COVID versus those who also were immune to OC43 but did not get COVID. Both groups had similar concentrations of antibodies to the N protein on the SARS-CoV-2, suggesting that this prior immunity is simply reactive and not protective. The study also found no significant difference in disease severity and hospitalizations between people who had prior immunity to seasonal coronaviruses and those who did not. If prior immunity doesn't affect disease severity, then there may be more to the story than we previously had thought. The cause of asymptomatic infections may be related to either the host or the virus itself. So far, we've been discussing how changes in the host could impact asymptomatic infection. Considering there's conflicting data in that topic, it may be useful to also talk about aspects relating to the virus, in particular incubation period and viral inoculum, which is the amount of virus that enters the host that may play a larger role in asymptomatic infection. The viral inoculum is an important factor in the severity of symptoms the host tends to face. In particular, if the host is exposed to a higher concentration of viral load, then the host develops more severe symptoms. If the viral load the host is exposed to is less, the symptoms are more mild. So this brought in the whole mask wearing idea, and the main reasoning behind it is that mask wearing tends to lower the viral load of not only the wearer is exposed to, but also those around the wearer. And so as a result, on average, people tend to experience milder symptoms, and so that goes to show that this can be linked to an increased rate of asymptomatic infection due to mask wearing. As people on average are better protected, the average amount of viral load they receive is greatly reduced, and hence reducing the risk of symptom-heavy infection. There's plenty of data throughout virology to show that the infectious dose of a virus correlates with disease severity. A study that infected individuals with the wild-type influenza A virus found that disease severity was strongly correlated with the viral inoculum of influenza. This has been corroborated in many animal models relating to both SARS and COVID-19. With the increase in global masking, we may actually be increasing the number of asymptomatic cases. Besides just being a protective measure to keep sick people from spreading the virus, it also acts as a filter that reduces the viral inoculum, likely reducing the potential severity of their sickness. This may even correlate with the incubation period of COVID-19. This graph here shows a negative correlation between incubation period and the time between symptom onset and discharge from the hospital. The difference in incubation period would be explained by the viral inoculum, suggesting a correlation between viral inoculum and incubation period. Now, another question that remains to be answered is the strength of immunity based on all these different factors.
So a lot could change toward the future. Most of what we're speculating about COVID-19 immunity could turn out to be wrong in just a matter of weeks. When we were creating our outline for this TED Talk, many new studies were published in preprint which contradicted some of our ideas. So we had to change the direction of our talk a bit, and I think that's really cool because it shows how fast the landscape of science is changing, and I think it's really interesting to watch it happen in real time. So let's talk about vaccines a little bit. Specifically, would vaccine immunity be stronger than natural immunity? This is an interesting question because natural immunity seems to be highly variable among different people. Depending on the severity of your disease, the length of your incubation period, and the viral inoculum, the robustness of your immunity could change. The current data with reinfections suggests that natural immunity may not be so strong, so the CDC actually recommends vaccination 90 days after being infected with the COVID virus. So in order to understand this a bit better, let's go briefly over the Pfizer and Moderna vaccines. Here's the efficacy graph from the Pfizer vaccine. And just to let you know, Moderna's efficacy graph looks quite similar to this. Here they're measuring the incidence of cases, blue is placebo and red is the vaccine group. And there was around a 94% reduction in symptomatic COVID-19 disease in participants who received the vaccine, but it's still unknown whether or not the vaccine actually prevents infection of the virus. I wanted to focus on the T-cell responses from both these vaccines. Both vaccines had a comparable CD4 T-cell response, but the main differences are in the CD8 responses. Here they're measuring interferon gamma activity among CD8 T cells as a percentage. A higher level indicates a stronger CD8 T cell response. At the 30 microgram dose, which is what Pfizer is going with, we can see that the value is around 0.25, but in Moderna's vaccine, there was a much lower CD8 response. The blue dots indicate interferon gamma activity among CD8 T cells as a percentage, and the peak at day 43 was much less than 0.2, which indicates a much weaker CD8 T cell response from the Moderna vaccine. Although it's unknown whether or not these differences are clinically relevant, we suspect that the Pfizer vaccine may have a longer lasting immunity to the virus as a result of stronger CD8 T cell response. Moving forward from COVID, I think we will have a much better plan for future pandemics in general. I believe more focus will be placed on discovering emerging viruses that have pandemic potential, and there will probably be more funding in researching these viruses as well. I also think that the new mRNA vaccine platform will dramatically change the impact of vaccines in the future. The RNA platform shortens the vaccine production process since it doesn't require actually synthesizing any proteins, making it much easier to produce large doses of vaccines in a small period of time. I wanted to briefly touch on other vaccines as well that are in development. Australia recently canceled their $750 million vaccine plan after it caused HIV false positives. Other vaccines, which are still deep in development, involve nasal sprays as a delivery mechanism, which will most likely be less effective than the needle, as shown in the influenza nasal spray vaccine. Overall, I think that the knowledge we gained from the immunogenicity of this virus in the future would be very useful in understanding and treating other diseases later on. Understanding the effect of asymptomatic transmission may also help us contain the spread of a virus in the future, hopefully preventing something like this from happening again. And before we end our talk, we want to bring up whether or not COVID will become endemic. Will we need a vaccine every year like the flu? Will COVID stick around for the rest of our lives? Coronaviruses don't seem to mutate like the flu, so unless something changes in the near future, I don't think we will need a yearly vaccine. But in terms of actually wiping out the virus from the population, herd immunity will be necessary, And considering that many Americans are unwilling to get vaccinated, I don't think we will reach herd immunity simply through the vaccine. But if the vaccine does turn out to actually prevent COVID-19 infection, I think that the prevalence of COVID will dramatically decrease in the population either way. But I still think it's too early to say whether or not COVID will become endemic, and we will just have to wait and see what happens in the future.